Hey, before we jump in, if you want to know how we can literally turn a gene off, go ahead and hit that like and subscribe button. It helps a ton. All right, let's do this.
Welcome back, everyone. Today, we're diving into something super cool in genetic medicine. It's called RNA silencing. Basically, we're going to talk about how scientists can find a single problematic gene and just turn it off.
So, in our last explainers, we talked a lot about adding genes, right? Gene therapy. But what if the problem isn't a missing gene? What if it's the total opposite? What if you've got a gene that's just doing way too much, causing all sorts of trouble? You can't just snip it out. So how do you tell it to just be quiet? How do you turn it off?
Well, the answer, and this is the amazing part, it comes from a process that's already happening inside every single one of our cells. It's this super elegant system called RNA interference, or, RNAi for short.
Okay, so to really get how this works in the real world, let's look at a problem most of us have heard of, high cholesterol. And to do that, we need to meet the villain of our story today, a gene called PCSK9.
All right, quick refresher. LDL cholesterol. You've probably heard it called bad cholesterol. And yeah, when you have too much of this stuff floating around in your blood, it's bad news. It can build up and lead to some really serious heart problems. Now, our liver, that's the organ that's supposed to be cleaning all this stuff out of our system.
Okay, so this is where our villain PCSK9 enters the scene. This gene is mostly active in the liver, and it makes a protein that has one job and one job only, to find and destroy the very things our liver uses to clear out that bad cholesterol. These things are called LDL receptors. Think of them like little hands on the surface of your liver cells, reaching out and grabbing the LDL from your blood. Well, the PCSK9 protein comes along and just chops off those hands. So you can see the problem, right? The more PCSK9 you have, the fewer hands your liver has, and fewer hands means that bad cholesterol just keeps building up.
So, if PCSK9 is the problem, the solution seems pretty obvious, right? We just need to turn it down. We need to find a mute button for that one specific gene. And that is exactly what RNA interference lets us do.
And the really wild thing is, this isn't some crazy new technology we invented from scratch. As this quote points out, RNAi is a fundamental process our cells use all the time to control which genes are on and off. We're not inventing a tool here. We're just borrowing a masterpiece that nature already perfected.
Okay, let's get into the nitty-gritty. This diagram shows the whole mechanism. Now, don't worry, it might look a little complicated, but we're going to break it all down. The key thing to remember is that RNAi is like a super-specific guided missile system. We send in a small piece of guide RNA, the cell's machinery loads it up, and then that machinery hunts down and destroys the exact message from the gene we want to shut down.
Let's walk through it step by step. First, we introduce a custom-made molecule called an siRNA. Just think of it as a perfect molecular mirror image of the message coming from our villain gene, PCSK9. Next, the cell's own machinery, this cool complex called RISC, grabs that siRNA, unzips it, and loads one strand as its guide. The weapon is now armed. Then, step 3. This armed RISC complex starts patrolling the cell, hunting for one thing and one thing only, the messenger RNA from that PCSK9 gene. And finally, step 4. Silence. Once it finds its target, bam! It literally slices the messenger RNA in half, destroying the instructions. That harmful PCSK9 protein never even gets made. The gene is officially silenced.
Okay, so we've got our weapon, right? This siRNA molecule. But here's the catch. This whole process has to happen inside liver cells. So the big question becomes, how do we get this tool specifically to the liver and not everywhere else in the body?
For a long time, this was a huge hurdle. It meant using these really complex delivery systems, like lipid nanoparticles or even specially designed viruses. But for the liver, it turns out, scientists found this incredibly elegant solution, a new key, if you will. And it's just a simple sugar molecule called GalNAc.
And this is where the genius comes in. The beauty of this is its simplicity. Turns out, our liver cells are just covered in these special receptors that are designed to do one thing: grab on to GalNAc. It's like they have a special lock, and GalNAc is the key. So, by just sticking our siRNA onto a GalNAc molecule, we basically give it a VIP pass straight into the liver. It's a natural, highly targeted delivery system.
Okay, let's put all the pieces together and see how this works in a real, actual, approved therapy. This is where the villain, PSK9, the silencing tool, siRNA, and that special delivery key, GalNAc, all come together in a drug called inclisiran.
So here's the whole story of inclisiran.
You've got the GalNAc molecule, that's our hook. It's attached to the siRNA, that's our payload. And that payload is designed to do one thing: target the message from the PCSK9 gene. A patient gets a simple injection, just under the skin. The drug travels through the bloodstream, and that GalNAc hook acts like a homing beacon, guiding it right to the liver cells. Once it's inside, the siRNA is released, and the whole RNAi process kicks off, silencing the PCSK9 gene. And the result? The liver stops destroying its own cholesterol receptors. Those little hands grow back, and the liver can get back to its job of clearing all that bad cholesterol out of the blood. Mission complete.
So, the big question is, does it actually work? And how well? The results are, well, they're pretty stunning. In the clinical trials, inclisiran caused an average drop in that bad LDL cholesterol of over 52%. I mean, just to put that in perspective, that is a massive reduction. It's the kind of drop that can seriously, dramatically lower a person's risk of having a heart attack or stroke.
And it's not just that it's powerful, it's how long it lasts. That's the real game changer here. Because we're messing with the instructions at the genetic level, the effects stick around for a long, long time. This quote from the trial data says it all. The cholesterol-lowering effect is sustained for at least 6 months. Think about that. Instead of having to remember to take a pill every single day, a patient might just need an injection twice a year. That's incredible.
So, look, this is so much bigger than just a new drug for cholesterol. This is a proof of concept for an entirely new way of making medicine. We've shown that we can create a highly specific, super durable off switch for almost any gene we want. And we have a precision delivery system to get it to the liver. So that leaves us with the big question, right? This targeted off switch works for cholesterol. What other disease-causing genes could we go after next? The possibilities here are they're truly revolutionary.
It's a really, really exciting time in medicine. But that's all the time we have for this one. Thanks so much for hanging out with us to break it all down. We'll see you on the next one.